it's a fact that some people don't like that resveratrol is the most debunked longevity molecule ever. Resveratrol does not increase lifespan in laboratory animals on average. So right now, my confidence is 100% that you can improve your health span and likely your lifespan through lifestyle modifications.
I traveled to Seattle to talk with Dr. Matt Kaberlein, who's one of the world's leading biogerontologists and longevity researchers. He's been researching aging and lifespan for over 20 years. I asked Matt questions from my audience about different longevity protocols, supplements, and lifestyle. The first question is, so recently Bryan Johnson stopped taking rapamycin. What are your thoughts about that?
Yeah, well, specifically with respect to rapamycin, I mean, I think first of all, the honest answer is nobody knows whether rapamycin slows aging in humans or whether or not there are going to be significant benefits from rapamycin. I don't know. Bryan Johnson doesn't know. Nobody knows because we don't have randomized clinical trials yet, at least for disease prevention, health promotion, right? They're all limited to organ transplant patients or cancer patients, things like that. So nobody knows. Bryan Johnson's specific reasons, as I understand them, for stopping taking rapamycin. So it's his personal decision. He has every right to do whatever he wants with respect to his own health. But his rationale, as I understand it from what I've seen in the public domain, is nonsense, right? So he's basing it on one study that used biological age assessments that don't work. And in that same study, they found that exercise and quality diet doesn't have a positive impact on biological age. So using that as a reason to stop taking rapamycin, because rapamycin also did not have a positive impact on biological age, is just dumb in my view. Like, it's nonsensical. Now, he's also said that some of his biomarkers may have been perturbed around lipids and glucose. That's possible. Those are side effects that are associated with rapamycin at high doses in organ transplant patients. They have not come out as significantly associated with rapamycin in people using the doses that he's reported to use or that have been used off-label. So I find it unlikely, because we've looked and other people have looked, I find it unlikely that rapamycin had that effect on his biomarkers, but it's possible. It's also, there's no way to know for sure because he's doing like 100 other things. So again, the idea that you can attribute that to rapamycin is not rigorous and not scientific, which is one of the many reasons I say that he does not take a scientific approach to those kinds of things. So again, I'm not suggesting anybody take rapamycin, but I also, if you are taking rapamycin and you feel like it's benefiting you, I would not pay any attention to Bryan Johnson, would be my advice.
Yeah, I guess it's like, experimental therapy for life extension. Like, we don't know if it's going to work in humans. Obviously, it has been shown to work in animals.
Absolutely.
Which would be, in my opinion, like a greater degree of certainty that it would have effects in humans than the biological age clock in that which he saw.
Especially when the things we know work in humans didn't come out with a positive effect in that same study, right? So again, it's obviously, I'm a little bit frustrated with the fact that this gets headlines and it's just not helpful for anyone in my view. But having said that, you're right. We have no idea whether rapamycin increases lifespan in people. It does in laboratory animals, and quite robustly. I think what we have learned is that it's at least some people see a reduction in chronic inflammation and, at least anecdotally report benefits like reversal of hypertrophic cardiomyopathy, reversal of autoimmune disease, in my own case, reversal of inflammation of the shoulder capsule, commonly known as frozen shoulder. So it hasn't risen to the level of randomized double-blind placebo-controlled clinical trials. It's more like a series of case reports. I do believe some people get benefits. I do believe some people experience side effects. The only side effect that seems, again, statistically significant is canker sores. About 15% of people using rapamycin off-label get canker sores. There may be a slight increase in risk of bacterial infection, although that hasn't risen to the level of statistical significance at this point.
Yeah, like one of the fears I've had in the past about rapamycin is that it suppresses your immune system and makes you lose muscle. But over the last few years, that has been turned out not to be the case, actually.
Certainly in laboratory animals, it's the opposite, right? You can actually boost age-related immune function and maintain muscle mass and prevent sarcopenia in rodents in the laboratory. And in people, again, it's anecdotal. We don't have the solid data yet. I should say there were 2 fairly large clinical trials that Joan Manick did with everolimus, which is a derivative of rapamycin. It works exactly the same way. And in both of those, they did see an improvement in vaccine response in older people. So probably rapamycin improves immune function, at least as measured by vaccine response in older people. We actually found in our study of 333 people using rapamycin off-label, lower incidence of severe or moderate outcomes from COVID-19 and a lower risk of symptoms associated with long COVID. No difference in risk of getting infected by COVID, but better outcomes in people using rapamycin. So I think that boost in at least antiviral immune effects is probably real. And there's a clinical trial that I believe the data has been collected. I haven't seen it yet for muscle function in elderly people that Brad Stanfield has gotten funded and has been running out of New Zealand. So, we'll see. The Pearl trial from Ageless RX showed a hint of improved body composition in women at very, very low doses. So I would say, if anything, the needle right now is leaning towards better outcomes in terms of body composition and lean mass in people. And certainly that's what's seen in the rodents.
Yeah, well, that's super interesting. Next question. Are more antioxidants always better? Like, there's research that antioxidant supplements can actually be harmful.
Yeah. So, I mean, the answer is obviously no, right? Nothing is always in biology, or very little. It's useful to appreciate the term antioxidant is sort of misused, right? So it's all relative to other molecules, right? One molecule can oxidize or reduce another molecule depending on their relative oxidation status. But most of the time when people are thinking of antioxidants, they're thinking about, supplements that you can take that are generally going to reduce or remove oxidation from other biological molecules. And the idea is that as part of a byproduct of metabolism, we produce things like reactive oxygen species, which we know can cause damage. In fact, one of the early theories of aging was the oxidative theory of aging, that aging was really driven by oxidative damage. And then that was modified to the idea that the oxidative damage primarily comes from mitochondria, right? From metabolism. So based on that, it is certainly plausible that if you could prevent that oxidative damage, that would be beneficial. And there's evidence to support that in experimental studies. The challenge is that oxidative signaling is also important for health, right? So we know that oxidation status can be used as a way to signal throughout the body and within the cells important functions. And so if you block that, that's actually detrimental. I think the challenge with antioxidant supplements, aside from the fact that we don't know exactly where the antioxidants are going to go and how effective they're going to be, is how to target the bad reactive oxygen species and not target the good reactive oxygen species. And we just don't understand that biology enough. So the answer is, too much antioxidants is going to be bad. And I don't personally, and I don't think anybody really has a good feel for at the population or individual level, what is the optimal amount right now.
I guess like mega doses is probably not a good
I
idea.
wouldn't do it. Yeah.
And timing probably matters as well.
And particularly where you target. There's some interesting data around what people would call mitochondrial targeted antioxidants, or mitochondrially targeted peptides that reduce oxidative stress. It's intriguing. It has not yet to the level where I would suggest people should go out and start taking these things necessarily. But the idea that you could specifically target the antioxidants to the cellular compartment that is most problematic makes a lot of sense. And so I think there are opportunities there. We just don't have those tools yet.
Yeah. All right. Next question. What percentage do supplements versus lifestyle play a role in longevity? So how much percentage?
Well, I mean, I would definitely say that lifestyle is far more important than supplements, at least in terms of what we know is going to work. I think the challenge with supplements is we don't know what the right supplements are or how effective they could be. So right now, my confidence is 100% that you can improve your health span and likely your lifespan through lifestyle modifications. And my confidence is less than 10% that you can do that through supplements. And even if you could, I think it would be incremental compared to lifestyle. So lifestyle is by far the best opportunity to improve health span and probably lifespan for the vast majority of people.
What would be like the ratios? Like, do supplements give you 5% of
I mean,
health?
again, I don't know is the answer. I'm a scientist, so I have to say I don't know. My guess is, it depends on the person. So I would put supplements in a couple of categories. I think there are supplements where we can measure deficiencies or where we know most people are deficient. And if you're deficient, supplements can have a relatively large impact on at least mortality risk and risk of certain diseases, right? So, things like vitamin D or omega-3s, where there's pretty good evidence that if you're deficient, there's a higher risk of a variety of different negative health outcomes.
Right.
But then so I would say if for people who are deficient, supplements could have maybe it's 80% lifestyle, 20% supplements or something like that. But when we start getting into the more, questionable stuff, NAD precursors, spermidine, urolithin A, alpha-ketoglutarate, right? I'm just picking those out of air, and I'm not saying there's no value to those. They're speculative. And the nature of speculation is I have to guess what the likelihood is that they're going to work. I would say it's like 95% lifestyle, 5% supplements, maybe even more towards lifestyle. But that more comes from confidence that lifestyle is going to have a big effect.
Yeah, I agree. Like, even if there are like some supplements that have some effects, they're not going to replace exercise and good sleep and diet.
They may supplement those things, right? Like creatine's a good example, right? I mean, I think for a lot of people, creatine can have some benefits. I don't know if it's a big benefit or a small benefit. My guess is it's still relatively small compared to lifestyle. But I think especially if you're practicing a healthy lifestyle, having a supplement like that can give you added value in the context of practicing a healthy lifestyle.
Yeah. Okay, next question. Other than rapamycin, which compound is most likely to extend lifespan?
That's a good question. So I'm pretty bullish on SGLT2 inhibitors. So these are things like canagliflozin and pagliflozin. They have come out of the mouse intervention testing program as increasing lifespan in mice. There's human correlative data from UK Biobank that people taking these drugs have a lower risk of all-cause mortality compared to health-matched people who are not taking those drugs. They're anti-diabetes drugs. Basically, my understanding is they work by causing you to pee out more glucose from your circulatory system. So they seem pretty promising. I think estrogens and estradiols are really interesting. They also fit coming out of the interventions testing program in mice as increasing lifespan in males. And again, UK Biobank data seem to be associated with lower all-cause mortality in females. That would probably mostly be in the context of hormone replacement therapy in women, because not a lot of men have been put on estrogens or estradiols, but also maybe birth control. So I'm not suggesting that women, who are not perimenopausal or menopausal go out and start taking hormone replacement therapy. I don't think we're there yet, but the biology is intriguing. The thing is with the estrogens and estradiols, we don't understand the mechanism yet. So it's hard to know. I'm certainly not suggesting men run out and start taking these things, but I'm interested to see. So those are two classes that I would say have both preclinical and human data suggestive of lower all-cause mortality. And certainly I would put, again, I don't know about lifespan, but I would put hormone therapy, particularly in women, although I also think in many men, as a major health span booster. So we don't have longevity data yet, but in terms of impact on health span outside of lifestyle, I would probably say hormone therapy is maybe the next biggest thing that a lot of people can benefit from.
What about something like acarbose?
Yeah, acarbose is interesting. That also comes out of the interventions testing program as one of the first things after rapamycin to show a robust effect. Also only in males or mostly in males, and we don't really understand the sex differences there. The human data on acarbose, we don't have we don't have the sort of UK Biobank level, and At least this is my understanding, in the context of a Western diet, acarbose is not really well tolerated because of gastrointestinal upset, flatulence, things like that. So, um, I'm a little bit, I mean, it's interesting. I'm a little bit less bullish on the translational application of acarbose than SGLT2 inhibitors, probably working through a similar mechanism. Acarbose, so again, acarbose is also an anti-diabetes drug. I mentioned the SGLT2 inhibitors cause you to basically pee out more glucose. Acarbose probably works by causing you to take up less glucose or other sugars that get converted into glucose because it's an inhibitor of an intestinal enzyme involved in breaking down complex carbohydrates. So as a class, I think anti-diabetes drugs tend to promote longevity in laboratory animals, and certainly in Western populations, metabolic dysfunction is a hallmark of aging that probably drives a lot of age-related processes. So anti-diabetes drugs, metformin being the classic example, are often thought about in the case of longevity. Maybe it's worth talking about metformin as well in this context. I'm not particularly bullish on metformin in metabolically healthy people. I think, again, in people who are diabetic or pre-diabetic, metformin is a really good diabetes drug. Unlike acarbose and SGLT2 inhibitors, metformin does not appear to increase lifespan, or at least if it does in mice, it's a very small effect. It did not come out as positive in their intervention testing program. And the early data that people taking metformin were at lower risk of dying has not been replicable. So if you're diabetic and you're taking metformin, you're at lower risk of dying than if you're diabetic and you don't take anything. But in people who are not diabetic, it doesn't seem to have replicated that there's any real benefit in terms of mortality. And we're learning that metformin may blunt some of the positive benefits of exercise. So if you're not diabetic and you have a healthy lifestyle, I think metformin is probably a net negative there.
Yeah. Next question. What's your most unpopular opinions on lifestyle and supplements? Like controversial takes.
I don't know if it's unpopular. So around lifestyle, I don't know that I have any unpopular opinions. I mean, I sort of align with what I think is the consensus view around, eating a generally whole foods diet. Plant-based, I think, is a good idea. I like meat. I eat meat, but I think largely plant-based. I tend to align towards moderate to higher protein consumption, limit alcohol, avoid added sugars, right? That I think most people who are informed are going to land there. Exercise, mix of cardio and resistance training, sleep quality, relationships, the pillars of health. So I don't think there's anything, I don't think there's anything controversial there. In supplement world, again, I tend to be pretty skeptical. I don't think it's controversial. It's a fact that some people don't like that resveratrol is the most debunked longevity molecule ever. Resveratrol does not increase lifespan in laboratory animals on average, right? There's been a meta-analysis, it's not even my data, of like 50 or 60 studies, and the average effect is 0. I'm sorry, that's a fact. If people don't like it, too bad. So, it's just reality. And yet you still hear, certain influencers out there peddle the nonsense that resveratrol is a longevity drug or an anti-aging drug. Other than that, I don't have super strong opinions on most supplements, other than I tend to be maybe more skeptical or less enthusiastic than other people out there.
Yeah, just more waiting for more data.
Yeah. And I tend to also, my view is that I tend to maybe be a little bit more conservative when it comes to adding a supplement to my personal protocol, because I take the view that the human body is a really, really complicated piece of machinery, or at least that's a good anecdotal way to think about it. And if you consider any complicated piece of machinery and you go in there and you start taking screws out or pulling wires out at random, the chances you're going to make it work better are less than the chances you're going to break it. And I kind of think that's what a lot of people are doing when they start stacking 10, 20, 30 supplements. They're just throwing a lot of stuff at it, and they assume that there's no risk. But the reality is there is a risk. It's probably small in individual supplements. But when you start to stack a lot of stuff, again, my view is, unless you have a very, very good rationale and hopefully a good understanding of the biology that you're perturbing, the likelihood you're going to get a better outcome is less than the likelihood you're going to get a worse outcome.
Yeah, I guess you need to know if it's working or not either.
Well, and that's where I think having good biomarkers is really important.
Yeah.
And I think we've got reasonable biomarkers for general health, right? What we don't have are good biomarkers of aging, longevity, right?
Yeah.
Yet. And we can talk about biological age clocks, but at least the ones that are available to consumers now, in my view, are not sufficient quality that we can use them to guide interventions.
You got very conflicting results from
so I did my own. Maybe that, I mean, this isn't a supplement related, but that is maybe a controversial opinion. Again, I get that it's controversial. I don't see the controversy because I'm just pointing out data. Like, you know, you can choose to be data driven or not. I choose to be data driven. And so I did an experiment where I tested 4 different biological age tests from 4 different companies. These are all consumer facing. Anybody can go on the internet and buy these things, right? And I did them in duplicate. I wish I'd done triplicate or 4 of each ones, but you know, as you know, these tests are anywhere from $300 to $600 a pop, right? So even doing 8 is fairly expensive. So I did 4 tests from 4 different companies in duplicate, same biological samples taken on the same day. I was 53 3/4 years old at the day that I did this. The results I got back ranged from low 40s to mid 60s, right?
Right.
Now, if I took the average of all 8, it was almost spot on my chronological age, but the variance was huge. And so, you know, and to be fair, again, I only had duplicates. Most of the companies, the 2 duplicates for the same test were within 3 to 5 years. That's still a pretty big range, but, you know, relatively close. One of the companies was 20 years apart. But I think the bigger point is that we have no idea, first of all, what my true biological age is. So we can't know what the accuracy of these tests is, but we can know the precision is shit, right? The noise is bigger than the signal. And I think that's the problem, is none of these tests have been, you know, validated by any regulatory body, any independent, you know, consumer watchdog organization. It's just these companies saying our tests measure biological age. I don't know why people would trust them, but they don't. Or if they do, we don't know which ones do. And so I don't think we can use these consumer-facing biological age tests for anything right now other than enriching the people who are at these companies. They're really useful as research tools. And so I don't want to blast the idea that we can measure some aspect of biological age. Again, I try not to, but I've even been using that term sloppily, you know, just in the last few minutes. We can measure aspects of biological age in the laboratory setting. My concern is the rush to market and selling these to people when they aren't ready for prime time.
Do they use like the different clocks in the laboratories? Or
That's right.
Okay.
Yeah, and so maybe it's worth just briefly talking about what's actually being measured in most of these. So first of all, people should appreciate you can measure biological age a bunch of different ways, and everybody will call it biological age, but none of them are measuring biological age as a true thing. What they're measuring is usually a correlation to mortality risk, right? And so we can do that with things like LDL, HDL, insulin, glucose, right? We can come up with a set of those markers that everybody gets on their blood test and predict your risk of dying in the next 10 years. Now, it may or may not be a good prediction, but we can predict it. That's what people are calling biological age clocks. It's a correlation to mortality risk. So in most of the consumer-facing biological age clocks, they're not using those blood-based biomarkers. They're using something called DNA methylation. So these are chemical modifications on our DNA, and there are hundreds of thousands of them in our genome, and they change over time. And importantly, they determine which genes in our genome get turned up or turned down in terms of expression. So it's a connection to biology that's really important to aging. People have found that the patterns of these methylation marks change with age. And you can look at, you know, depending on the clock, anywhere between a few dozen to 150-ish of these specific methylation marks at specific locations in the genome. And you can identify a correlation with chronological age within a year or two in the laboratory. in principle, these companies can do exactly that same test and get a correlation to chronological age if you send them your blood. But we don't know if they're doing exactly the same test or whether it's done well. And then those are the first generation clocks. The second generation clocks, which people may have heard of, are instead of being based on chronological age, trained on chronological age, it's a different set of marks that are correlated with 3-year, 5-year, 10-year mortality, your risk of dying, or your risk of developing specific age-related diseases. Same principle, though. It's a correlation to those things. And those things, mortality risk, is correlated to biological age, right? It's not a perfect correlation, though. It's not the same as biological age. So really what these tests are measuring is a correlation to a correlation, but they're calling it biological age. That's part of, I think, the lack of clarity about what we actually mean. So again, I think the take-home message, though, is these can be very powerful in the research world. And I think we'll get to a point where they can be useful, say, in clinical trials, in predicting whether or not a new drug, a new intervention is going to lower risk of mortality. And when used responsibly, if we can convince regulators that this is actually a really good prediction of your risk of dying, you could do a clinical trial and get a drug approved. It would be what's called a conditional approval, potentially, based only on the test. But we have to convince the regulators these things actually work. And this is another potential problem to the companies rushing these things to market. When the regulators realize they don't work, because the ones that are being rushed to market don't work, it's going to be harder for responsible people to convince them, oh yeah, they actually do work because we're doing it right. Maybe the last thing that's worth commenting on, because I know you've talked about this as well, you can also use a different set of methylation marks that are correlated with the change in health over time. This is the pace of aging, the Dunedin pace of aging or rate of aging. So there's a different set of methylation marks across a population level study that had been shown to correlate with whether people stay healthy over time or have a decline in health over time, or maybe even their health gets better. And so based on those marks, people developed a different clock that is supposed to measure your rate of aging, your pace of aging. Again, it's a correlation to health outcomes, and a change in your health is correlated to a change in your biological age. So it's still a correlation to a correlation, but that's really valuable if it works, because then you can do experiments and say, what happens if I stop drinking alcohol? Does my rate of aging go up or go down? And be informed by that. So again, that's really, really exciting. And it seems to work in the laboratory, in a research setting, or maybe not, I shouldn't say laboratory, in a academic controlled study, could be of people. But we have no idea whether these companies that are telling you your pace of aging, whether that is meaningful or not, because again, it's not, there's no quality control or external validation.
Yeah, I tend to agree. Yeah, like there's a lot of noise with those tests. I want to take a quick break to let you know that you can now get my new book, The Longevity Leap, on Amazon. It contains 24 chapters ranging from the biology of aging to all the major chronic diseases such as heart disease, kidney disease, neurodegeneration, and I also cover over 70 clinically relevant biomarkers for chronic diseases and their optimal ranges. You can get the book from the link in the description. Next question, peptides for longevity. Do you have any opinion about that? So like BPC, GLPs as well?
Yeah. So first thing to say is I think people lump peptides together, but that's kind of like lumping drugs together, right? Peptides are just bioactive molecules that have a certain type of chemical structure based on the fact that they're a combination of amino acids. That's all peptides means. So they can have all sorts of different effects. And so it's important for people to appreciate, you wouldn't necessarily put rapamycin with aspirin with a PDE5 and with Viagra, right? They all can do different things. Peptides can also do lots of different things. They are biologically active. Our bodies produce many peptides. So you can categorize peptides based on whether they're naturally occurring or new drugs that people have synthesized, right? And I think it's plausible that naturally occurring peptides are going to be, like BPC 157, are going to be less likely within a physiological range to have really negative outcomes. Whereas if you're taking a new peptide that our bodies have never seen, they could be much more likely to be much more potent and have negative outcomes. course, there's going to be exceptions to that. Okay, so all of that is groundwork. I'm not aware of any peptides yet that have been shown to slow aging or increase lifespan in laboratory animals.
Right.
Do I believe they could? Absolutely. Do I believe peptides exist that will slow aging? Absolutely. I just haven't seen data on any of the popular ones.
Yeah.
I think GLP-1 agonists are super interesting. So Wegovy, Mounjaro, things like that people have heard of for anti-obesity. I think they have a really good chance of increasing lifespan in laboratory animals. The thing I'm really interested about there is it through, if it does increase lifespan, is it through a caloric restriction mechanism, or is it through a mechanism independent of caloric intake? When people first started talking about this, I was of the opinion that sure, they'll increase lifespan, but it's just going to be caloric restriction.
Right.
As I've seen more and more data on targets of GLP-1 agonists outside of limiting appetite or food intake, I'm becoming more intrigued by the possibility that they could actually have positive health benefits through other mechanisms. I would love to see that experiment done. I have not heard of anybody doing that experiment yet in laboratory animals, but I'd love to see it done. And in people, I think, again, there's clear evidence that GLP-1 agonists can have impacts on a variety of age-related diseases. That could all be because you're reducing obesity or taking people from, the obese range down to the overweight range, or overweight to normal health range. But it's also possible that there are other impacts. Again, speaking about GLP-1 agonists, it's worth, I think, emphasizing that unfortunately, the way those things were rolled out to people, a lot of people were not given sufficient education around the importance of eating a quality diet when you're taking these drugs and doing some sort of resistance training. A lot of people lost a lot of lean mass in addition to fat mass. And I think what we've learned is when that happens and people come off these drugs, they tend to gain much of the weight back, but they don't get the lean mass back. So what we may be doing if people are not eating a healthy diet and practicing resistance training, is increasing frailty and osteoporosis in the elderly population. So just important for people to really understand. Great drugs, very powerful, they work, and you need to take steps to ensure that you maintain or actually improve your body composition when you're taking them. I think that's gotten better. The last thing I'll say about that is also pay attention to hormones when you're on these things, because anytime you're in a calorically restricted state, your hormones can become perturbed. Okay, that's GLP-1 agonists. The other ones, this is an area where I need to do a deep dive, and I've been meaning to for a while, to really take a close look at the common peptides that are out there. I think BPC is super interesting. The papers I've read in rodents, really interesting effects, lots of anecdotal stories. Will it generally work for people as a regenerative peptide or a healing peptide? I don't know. It would be great to have clinical trials. The growth hormone secretagogues, again, really interesting. I wonder, is that any better than just taking growth hormone? I get you can do hand wavy sort of explanations for why it might be. I haven't really seen anything that makes me believe that. I also haven't seen a lot of data on how effective these things are at increasing growth hormone. And honestly, the data on growth hormone as a longevity drug is pretty weak. This is getting a little bit in the weeds, so we don't need to go there. I will say Greg Fahey and his TRIM protocol, which includes growth hormone, is intriguing. This is a thymus regeneration protocol. But outside of that, I haven't seen a lot of evidence that the growth hormone per se is good for longevity. And then there's a variety of other peptides that potentially target mitochondria and other things. Very early, very experimental. It is interesting how many doctors in this space are comfortable prescribing these things with very, very little data.
Yeah, well, one of the questions is actually about the TRIM protocol.
Yeah.
So you can explain
Sure.
what it is.
Yeah. So I actually have an episode on the Optispan podcast for people who want to get a deep dive into this. I sat down with Greg about a month ago down in LA. So the TRIM protocol is based around the idea that relatively early in life, so adolescence or post-puberty, early 20s, there's a pretty dramatic decrease in thymic function. And our thymus is important for among other things, producing T cells, which are really important for the function of our immune system. So the idea is that because the thymus declines pretty dramatically, and then there's this slow but steady decline as we get older, this is contributing to immune dysregulation, both a decline in immune function and an increase in what's called sterile inflammation, or something like autoimmunity. So Greg came up with an idea for how to restore thymic function in people who are 40s, 50s, 60s, 70s. It's called TRIM. And the protocol is basically growth hormone, DHEA, which is a precursor for testosterone and other hormones, also declines with age, and metformin. And the rationale, as I understand it, is the growth hormone is really doing the action here. So the growth hormone does a couple of things to the thymus. So one of the things that happens during what's called thymic involution, this dramatic loss in function, is there's an infiltration of fat into the thymus. We see this in other tissues as well, but it happens early in the thymus. So growth hormone reverses that and at least causes the volume of the thymus to grow, right? Now, is it actually new cells, new stem cells? I don't know that we know that, but you can see it by MRI. You can see an increase in volume of the thymus. The DHEA is sort of in there. Not a great scientific rationale, but in part because DHEA goes down with age, and it may have some benefit for thymic function and maintaining hormonal balance. And again, we talk about this a little bit in the episode for people who are interested. The metformin is in there because growth hormone can have a negative impact on insulin sensitivity and glucose homeostasis. So metformin, again, anti-diabetes drug, this is to sort of offset that. I asked Greg about this. There's no data. I am intrigued by the idea that maybe SGLT2 inhibitors could substitute for metformin, counteract maybe some of the bad stuff about metformin. If SGLT2 inhibitors have a longevity effect on their own, also give you that. But there's no data on that. So what Greg has shown is in a number of small but intriguing human studies, evidence that the TRIM protocol over several months can improve thymic volume, reduce the fat infiltration, and probably improve T cell function and immune function, although that hasn't been measured in as much depth as at least I would like to see. They also have some evidence that the epigenetic clocks that we were talking about earlier show what would be perceived as improvements in biological age or reduced risk of mortality. So I would say pretty intriguing data, and it certainly makes sense that improving thymic function in older people is going to be generally beneficial. So I personally am at some point going to give TRIM a try. I just need to figure out the logistics of the MRI. I want to make sure I measure it, right? I want to know like what actually happened. So the logistics of the MRI analysis, make sure I get that done before, get, at least some biomarker panel that's going to tell me about inflammation and immune function before, and then do the same thing afterwards.
Right. Would rapamycin have any effect on the thymus?
Yeah, I asked Greg that. He doesn't have any data.
Okay.
I don't know the answer. I don't know. I mean, I think it's possible that you would also see a restoration, at least of thymic volume and a reduction in fat infiltration from rapamycin. But it's really speculative. That's just based on some of the data on rapamycin effects on fat metabolism from laboratory animals. And then there's some intriguing data both in rodents and in humans on rapamycin treatment increasing volume of the hippocampus and the caudate nucleus. So it's in the brain, very different, right? But by MRI, at least in people who are ApoE3/4 carriers. So ApoE4 is one of the biggest genetic risk factors for dementia and heart disease. So they tend to have an accelerated decline in brain volume in those regions. And there's 2 studies now that have shown, or at least strongly suggested, reversal of that reduction in brain volume, as well as improved cerebral blood flow. So I'm intrigued by the possibility, yes, that rapamycin could have an impact on thymic function, but no data that I know of.
Yeah. But does it like slow down immunity in a sense? So the decline?
I mean, So what we know from laboratory studies and then the human studies I alluded to earlier from Joan Manick is that functionally you see an improvement in vaccine response. So there's definitely a change in age-related immune function decline. And you see very potent reductions in chronic inflammations in mice across many different tissues and organs. And again, anecdotal data in people with, autoimmune or chronic inflammation that they have an alleviation of symptoms of inflammation with off-label use of rapamycin. So I think it's a reasonable hypothesis that rapamycin would have a positive impact on immunosenescence in people. I don't think we have any data directly addressing that at this point.
Gotcha. Next question, is IV NAD pure hype? Intravenous.
I mean, I would say it's a little bit better than pure hype. Again, I've talked to docs who do it regularly. They say some of their patients have some pretty significant improvements in how they feel. So I wouldn't want to say there's nothing there. Again, I'm not aware of any data, from rigorous studies showing benefits in a population of people or across a population of people from IV NAD.
Right.
Again, the hypothesis behind this is that NAD levels decline with age, and that by restoring NAD, that's going to be beneficial. That hypothesis or that idea that NAD levels generally decline with age is also not strongly supported by the literature. Again, there's evidence there, but that has been sort of overstated by people who have a vested interest in promoting that model. Often they're selling NAD precursor supplements. So I think my gut feeling is some people, particularly those who have significant mitochondrial dysfunction or metabolic dysfunction, do indeed have perturbed NAD homeostasis. And that can be reflected as either a decrease in NAD itself or a shift in the redox state of NAD towards NADH. Those people may benefit and feel better from NAD drips or from NAD precursors. I think maybe the last thing to say on this is there's a variety of ways that people are trying to boost NAD. So the NAD drips is one. NAD precursors that are taken orally, NMN, NR are the two expensive ones. Nicotinamide, niacin are the two cheap ones. All of them, in theory, if you take enough, can boost your NAD levels. And then there's a sort of a new sort of movement towards sublingual NAD or sublingual NMN. All of those, in theory, should increase circulating NAD levels. And so I don't know of any reason to believe that one is necessarily better than the other from that perspective.
Yeah.
I've never done NAD drips, by the way.
Yeah.
I'm sort of intrigued. I'm kind of like you, where I like to try stuff and see, do I feel anything? But I haven't tried it yet.
I haven't tried either. I've heard it's like can be uncomfortable because it's like several hours long, and you can do it fast if it's more painful.
I mean, this, I don't know if this is on your list or not, sort of related just because it's IV related and takes a few hours. That one thing I am becoming more intrigued by is therapeutic plasma exchange, right? The idea, and this is based on the parabiosis experiments in mice, that there's bad stuff that accumulates with age in our circulatory system and good stuff in young animals. And if you connect the circulatory system of a young mouse and an old mouse, this is an oversimplification, but I'm going to say it anyways because it sounds good, and it more or less fits the biology. The old mouse becomes younger, the young mouse becomes older, right? So therapeutic plasma exchange is kind of doing half of that equation, right? It's taking some of the bad stuff out of circulation. I'm becoming more convinced there's real potential benefit there, both in the context of aging, but also maybe in the context of things like, microplastic exposure or heavy metal exposure, right?
Yeah.
So I think that's interesting. I also haven't tried that yet, but that's definitely on the short list of stuff I want to try.
Yeah, me too. Like there's even like this EBO, which is like you do like a dialysis and filter the blood, and then you like shoot ozone and UV light into it as well to.
Yeah. That sounds a little more sketchy to me, but I mean, again, maybe
principle is.
It also comes from a little bit of lack of knowledge of what the research actually shows. Yeah.
Yeah. Well, the EBO is usually used for like autoimmune conditions and those kind of things. Yeah, next question. Does low IGF-1 increase longevity, and is low IGF-1 bad for the brain?
Yeah. So low IGF-1 during development, and let's just start with non-human animals first, right? So mice, rats, and dogs. I think it's pretty clear that low IGF-1 during development leads to smaller body size and increased lifespan on average. Every individual is different, but on average in a population, yes, it slows aging, seems to slow aging. In people, it's a little bit less clear, but there is data from studies of people who have mutations in, I think it was a growth hormone receptor, but that leads to lower IGF-1, that they are at lower risk for cancer, heart disease, metabolic disease, despite even having more adiposity, right? So they're smaller of stature. They don't live longer, but it's interesting because the increases in cause of death come from alcoholism and accidents. So lifespan doesn't change, but risk of at least some age-related diseases does change. So I think it's plausible in people that biological aging is slowed when you have lower IGF-1 levels through development as well.
How low? Like, because what you see from some of these observational studies is that if it's like too low or like below 100, then it can increase risk.
So again, I think you have to be clear. I'm talking about people who are naturally low in IGF-1 from gestation through development their whole life. So they probably have genetic variants that make them low in IGF-1. When you look at cross-sectional or even longitudinal population data in adults, that's a very different situation because those people presumably or may have gone through development with much higher levels. So they could still be large in stature or small in stature, but to have an IGF-1 level that doesn't match their stature, which is determined by IGF-1 levels in development. So, and I think that's important because, there are people who are thinking about therapeutic strategies to lower IGF-1 in adulthood with the idea that it will slow aging. As far as I know, there's only one study in mice that looked at this. It was a study that Derek Hoffman at Einstein did several years ago now. And they had a modest but statistically significant increase in lifespan. I want to say in male, but not female mice. I might be backwards on that. But nowhere near the magnitude of effect that you get from having low IGF-1 during development. And that probably was primarily an anti-cancer mechanism. And this is a place where, in general, I think studying aging in mice and then extrapolating to humans is reasonable. It's certainly much more reasonable than what people have done in mice with cancer or heart disease and artificially causing the disease. Mice age very much like people do. But mice in the laboratory don't tend to die from frailty, sarcopenia, fractures. They do get some of that, but it doesn't kill them. They tend to die from euthanasia, but usually the euthanasia is downstream of cancer. And certainly all mice will die with cancer. So it's a little bit different than people. I think the IGF-1 benefits, the lower IGF-1 benefits in adult mice primarily come from anti-cancer. So then the question is, will that extrapolate to people or dogs? And there's a company called Loyal that is moving forward with a drug that will reduce IGF-1 in adult dogs. We just don't know. One of the arguments you'll hear for a low protein diet in people is that it will lower IGF-1 and thereby lower cancer risk. The data on that is not particularly compelling. And again, there's a little bit of disagreement about what the epidemiological data actually shows. My take is that low protein, and that causes low IGF-1, but is the IGF-1 causing the downstream effects on mortality is not known. Low protein above about age 55 is generally a bad idea. You're at higher risk of frailty, sarcopenia, fractures, and death than eating a high protein diet. Obviously, that's a correlation, and it could be due to all sorts of other factors. High protein is also not great if you're eating an unhealthy diet. So if you're overfed and eating a lot of crap and eating high protein, that does increase your risk of cancer. So my view is the best bet is to eat relatively high protein, high quality diet.
Yeah.
That's independent of IGF-1, but worth saying. So, so does IGF-1 work differently in the brain? This is really interesting. Again, yeah, it does. I don't want to make a blanket statement about aging, but data from the Dog Aging Project is really intriguing here. So in almost every age-related disease that you can think of, and also for lifespan, small dogs age slower than big dogs do. Small dogs have lower IGF-1 through development. That's why they're small. And they live longer and seem to age slower than big dogs do. You can look at a bunch of different age-related diseases and functional declines, except for dementia. Dementia is the only one I'm aware of. There might be others. It's the only one I'm aware of that has come out of our data set so far where there doesn't seem to be any difference in dementia risk between small dogs and big dogs. So, and it doesn't scale with biological age. In other words, a 12-year-old German Shepherd has roughly the same dementia risk as a 12-year-old, I don't know, Chihuahua, right? Small dog. So that suggests that if IGF-1 is really driving the difference in aging rate in the rest of the body, the same relationship isn't there in the brain. And it's not as strong, but there might even be a hint of protection in the big dogs. And there's a hint of that in people as well. So yeah, it might be the case that having higher IGF-1 is better for brain aging. I think an interesting question is that effect due to higher IGF-1 during development, or is it due to higher IGF-1 during adulthood? We really don't know the answer to that.
So does the IGF-1 like help with neuron growth, or what does it do?
I don't think we know. Yeah, I don't think we know. Or at least I don't know. Maybe people who are experts in the brain or in dementia would have a better feel for that. It is interesting to me that one of the therapies, that has gotten some traction for dementia is intranasal insulin, and the insulin and IGF-1 signaling pathways overlap. So again, that may have nothing to do with this relationship, but it is sort of intriguing. There may be a connection there.
Yeah. I think it's with mTOR as well. Like mTOR is needed for neuroplasticity.
It is, yeah. I mean, again, you're right. The challenge with interpreting that as causal for anything to do with dementia is, it's really all about the levels of signaling through IGF-1 or mTOR. So, and where that comes from is if you completely inhibit mTOR, you take a very high dose of rapamycin, yeah, you'll block neuroplasticity, you'll block, new memory formation. But that's because mTOR is required for all protein synthesis and cell division, right?
Yeah.
So the question, and this gets back to like muscle function, which we talked about before, or lean mass muscle formation. If you turn down mTOR too much, you won't build new muscle. But is it possible that turning it down a little bit from where it's at, especially in the context of aging, is actually going to be net beneficial for muscle or for cognitive function or for memory formation? Yeah. I think that's why we see these benefits from rapamycin, because we're only turning it down a little bit and maybe transiently turning it down as opposed to just, dropping it down to the floor.
Yeah. Next question. Do you use AI and language models in your work?
I do. I use it a lot. Yeah. I mean, and it's growing, right? I mean, I think I'm certainly not one of these people who subscribes to the idea that AI is going to solve all of our problems, but I use it. We use it at Optispan, and we're thinking a lot about. So again, Optispan is really about creating tools, technologies to enable people to get on a better health trajectory, right? Writ large. So we have clinical programs, but we are absolutely trying to think about how do we use these AI tools to integrate lots of different data types, to get that back to people, to create interactive ways for doctors and patients to interact with their data. Lots of other people are thinking about this as well. I see really, really powerful use cases in the clinical application and what I would call health span medicine, or what Peter Attia would call medicine 3.0. Although I actually think that's medicine 4.0 when we add AI and geroscience on top of medicine 3.0. So yes, there are tons of really obvious use cases that are going to happen. You and I, within the next few years, will have a health span agent on our devices that we can interact with if we want to. Right. And it will function as well as most doctors. I'm very confident that can happen.
Yeah.
We're not that far away. So that's a really obvious use case for AI. I think in the broader, longevity geroscience space, AI is going to accelerate the pace of research everywhere, including geroscience and longevity. But I think we need much better data sets than we currently have in order for AI to really be a game changer when it comes to understanding the biology of aging, and being able to use that to impact lifespan and health span. I think our data sets right now are pretty bad. Certainly in the intervention space, our data set is woefully underrepresented for all of the different genetic or environmental, and I put small molecules under environmental things that we could test to see what impacts longevity. Tiny, tiny fraction. And I sort of, use the analogy that, garbage in, garbage out. Everybody gets that, right? You put garbage into the best AI tool, you're going to get garbage out. You put rapamycin and metformin in a carbosin, guess what you're going to get out, right? We need a better data set to know what are the most effective ways to impact longevity than what we've got right now. And then I would also add to that, we need larger human, what you sort of crudely describe as multi-omic longitudinal data sets. Basically, over time, in lots of people, measure everything you could think to measure.
Yeah.
Those are observational in nature, and they're very, very complex data sets, but that's the kind of thing that the new AI tools are going to be very capable of sort of teasing out what's important. So that's what I would like to see. The AI tools are going to keep developing. I'd like to see smart people in the longevity community actually put some thought into figuring out, as these things keep developing, how do we use them most effectively to really move the needle?
Yeah. Do you think like the AI will figure itself out things that isn't in the data, or like, because
I don't know. I'm less optimistic about that.
Right.
I kind of feel like the sort of foundational information needs, maybe I'm wrong. I hope I'm wrong, but I sort of feel like the foundational information needs to be there. And again, I sort of use this graphic sometimes in my talks where, I compare the hallmarks of aging, which are just a representation of how much we understand aging today, the biology of aging today. And I compare it to a map of the surface of the earth, in 500 BC, right? Which, I don't know if you've ever seen this map, Hecates's map, but it's got like a sort of, vaguely Europe-shaped thing in Asia that looks nothing like Asia, and Libya's
like a flat earth
10 times as and then you sail too far and you fall off the edge. So if you think about it that way, you could have the best AI tool in the world and you give it that map and say, show me the surface of the earth. It's not going to show you the surface of any earth that looks anything like what we know Earth looks like today. I sort of feel that it's the same idea if we give it how we understand aging today.
Yeah.
I'd love to be wrong, but that's the way I would guess it's going to go.
Yeah. Next question. How much protein for longevity? How much is it?
I mean, again, honestly, we don't know the answer, and it's going to be individual, right? So there's two things I would say. I would say for most people, the important thing, and it's going to depend on the quality of the diet, definitely, that you're eating. So I would say the best strategy for most people to adopt is what I would consider moderate to high protein. So anywhere from, and again, I'm just throwing numbers out, but anywhere from 0.6 to 1 gram per pound of body weight per day. But you got to be eating a high quality diet. If you're eating a crappy diet and you eat a lot of protein, you're going to increase your risk for cancer. Period.
So like sugar and
cut out simple sugars. Again, highly processed foods in general, cut out highly processed foods. We could have a debate. I mean, I had this conversation with Peter Attia recently, about protein bars, right? And his view is if you have really good ingredients and you can maximize sort of like the protein ratio per calorie, that's a good thing, right? So, and I think that's a reasonable thing. I still wouldn't say eat 10 protein bars a day, even if they're the best protein bars in the world. But I don't want to say never eat highly processed foods. But most highly processed foods have a lot of added sugars. Read the friggin' labels. I can't believe still today a lot of people don't look at labels. Like, cut out the simple sugars, right? Cut down the alcohol. Don't waste your calories on nonsense. So eat a high quality diet. Then I think high protein relatively, and definitely resistance training should be part of your exercise routine. That, to me, those three are really the strategy. And you can't really talk about protein in the absence of that. Might there be a slight increased risk of cancer from doing that compared to eating a low protein diet? Yeah, there might be. But I think by eating a high quality diet and exercising regularly, you probably dramatically reduce that increased risk.
Right.
And you're going to get lots of benefits in terms of muscle mass for metabolic health, bone density for frailty, hormonal health. Like, you're going to get all these other benefits that more than offset the if there is a tiny increased risk of cancer, the increased risk of cancer.
Yeah.
And again, I don't really I mean, you hear people do this all the time, and it's like, oh, I look at that guy and he looks 65, so his longevity protocol must not be working. I'm like, yeah, he's actually 75. It's working great. I don't like that sort of mindset. But the honest answer is, I know people who have dabbled with and still practice a very low protein diet. In general, they don't look very good and they don't seem very happy. So that's a generalization, but I do think that, if you look around in your own life and you see the same thing, that may be telling you something.
So no protein margaritas.
I never had a protein margarita, that's for sure.
I'm sure they're out there somewhere. Next question, what makes grip strength linked to health span?
Oh, good question. Yeah, I mean, I tend to think it's mostly just a reflection of overall muscle function and muscle quality, right? I don't think there's anything special about grip strength per se. I do think, having a good grip strength functionally for being able to do the stuff you want to do in your daily life is a good idea, but I definitely don't train for grip strength itself.
It's a byproduct.
Yeah, it's a byproduct of being strong, being able to lift weight. But like I said, if your grip strength is low, that will make it harder for you to do the stuff that you want to be able to do when you're 60, 70, 80 years old.
Yeah.
So be strong, I think, is the real answer there.
Yeah, like they find that even 20, 30 years in advance, if you have low grip strength, then you're going to have a hard time putting on clothes, getting off the toilet.
But that usually reflects a sedentary lifestyle and not having sufficient muscle mass throughout your entire body. Like, I think there are probably very few people who have sufficient muscle mass everywhere else and their grip strength is low.
Yeah, so I guess it's like, if you're otherwise with decent muscle and strength and in good health, you should have at least above normal grip strength.
Absolutely.
You don't have to train specifically for the grip strength.
Yeah, I've never been one of those guys that, has that gripper thing and does that, right?
Right. Although, I mean, I've used it. It's a very good exercise for the grip.
Yeah. It will increase your grip strength and your forearm strength.
Good for pull-ups, good for farmer carries, and good for arm wrestling if someone is interested. Next question, preserving women's fertility and AMH with HH. I guess that's like a marker of fertility or egg quality. Yeah,
so I'm definitely not an expert in female reproductive health. There are really, really smart people out there who are looking into this. I'll just preface it by that. So again, my immediate, answer to that is that, like almost any other, again, it's going to be dependent on your genetics and whether you're at high risk for, premature infertility or menopause or other ovarian syndromes. But in general, the same lifestyle factors that are going to have a positive impact on other aspects of age-related functional decline in other tissues and organs are also going to have a positive impact on reproductive health. So I know that's not a profound answer, but I think it's important to appreciate that's almost always going to be the place to start if you are concerned about a particular age-related functional decline or disease. And then you have to get into the more, either genetic risk factors or specific lifestyle risk factors that could have an impact. So I'm going to leave it there because, again, that's outside my area of expertise.
Gotcha.
It is interesting to me, and I view it as a positive development that the field as a whole is starting to pay much more attention to ovarian aging and female reproductive aging as both an early indicator of age-related processes and potentially a causal factor in differences in risk for specific age-related diseases in women. So I think there's a lot that's being done and a lot of work to be done there.
Yeah. Kind of follow-up question, what do you think of having children? There's data that having children later in life is associated with greater longevity in women.
Oh, okay. Yes. So there's two things to say there. So I thought you were going to ask about whether that has any negative consequences on the children, right? So yeah, I think my guess is the correlation between fertility, between childbirth later in life and longevity in women is mostly because women who are able to have children later in life have a later cessation of reproduction. In other words, their ovarian aging is delayed for some reason.
Yeah.
And so aging in the rest of the body is also probably proportionally delayed. That's the most likely explanation. Although, pregnancy itself does have an impact on a whole bunch of physiological functions. And using these DNA methylation clocks that we talked about before, you can see changes in the DNA methylation clocks during pregnancy and after pregnancy that look like an increase in epigenetic age followed by, in most women, a decrease in epigenetic age. So is it possible that the hormonal and other changes that happen during pregnancy are impacting biological aging itself? Yeah, I think that's pretty plausible. And in some women, it may actually reset some aspects of biological aging to a lower baseline value than before pregnancy.
Okay.
So it's total speculation, but that could also be a real thing.
Yeah, so there's like it increases during pregnancy, the biological age
The
score.
DNA methylation, patterns associated with biological age,
But then would you predict that the more children you have, the faster you would age, right?
Well, again, the data that I've seen, in most women it goes up, but then after pregnancy it comes back down.
Okay.
And in some women it actually goes down below the baseline where they started. So yeah, if it only went up, or if it only went up and partially came back down, you would expect an acceleration of aging.
Right. Gotcha.
But I mean, obviously having kids can have all sorts of other impacts on your stress levels and aging, and so it's complicated.
Yeah, I think that is, having generally more children is associated with slightly longer longevity to a certain point.
Sure. And I mean, again, that could be for all sorts of reasons. It could be biological effects of pregnancy. It could be later, the fact that when these women can just have children later in life, their ovarian aging is delayed, their aging is delayed in their whole body, could also be due to the social component, right? And, having children who hopefully are going to be around when you're an adult and maybe even care for you, right? So there's all sorts of factors that could weigh into that.
Yeah. The last question is, when should we start giving rapamycin to our dog? So that's from my wife. We have a puppy who is
I'm not a veterinarian. I do not make veterinary recommendations. But hypothetically speaking, if you were going to give rapamycin to your dog, the idea would be you're doing that because you believe that it might increase lifespan and improve health span in dogs. So, I mean, again, no way to know. So what I can say is, from our studies in the dog aging project, they have all been in dogs that are over 7 years old and at least 40 pounds. So that's, about 18 kilograms, right? And that's because big dogs age faster than small dogs. So it has nothing to do with the idea that rapamycin is going to work better in a big dog than in a smaller dog. It's just because for clinical trial purposes, you need a rapidly aging population. So that's a reasonable thing to think about, right? That roughly biological age. So if you're a 40-pound dog, 7 or greater. If you're a smaller dog, might be a year or two later. But that's just a guess. What we can say for mice is that, rapamycin treatment starting around that same biological age. So a 60 to 65-year-old person, maybe 55-year-old person, to a 7, 8-year-old dog, increases lifespan pretty robustly and improves a whole bunch of health span metrics. So that would be my sort of gut reaction for, and this is all really based on, I think maybe it's worth saying explicitly, what's the underlying sort of calculation here, right? It's always a risk reward. So what's the reward from starting when the dog's a puppy or the kid's a teenager versus the reward of starting when the dog's older or you're 50, 60, right? The difference in reward, the way we understand it now from mice, is pretty small. You get pretty much the same benefits starting later in life that you do if you start the mice very young. So then what's the difference in risk? Well, certainly if this is before puberty, obviously there's lots of risks that would go along with that. And I think the idea is the longer you're taking any intervention, any drug, the more likelihood that side effects could happen and compound. So again, with rapamycin, given that we don't completely understand the risk profile, I think we've learned a lot more. We still don't completely understand it. The risk is lower if you start later. So that's kind of where I land, both for off-label use in people and if you're going to do it in dogs.
Yeah. Our dog is 1.5 years old, and he's like 10 pounds or something, like 4 kilos.
Yeah, so pretty young still,
so
right?
he has a long time to go.
Yeah, which, that you're gonna most likely have that dog for 15, 16, 17 years, right? So
yeah. One interesting thing, like so he got car sickness, so we were giving him some of this nausea medication, which is like some antihistamine, and the ingredient was meclizine.
Yeah.
And actually, I found that there was one of the ITP ingredients
that came out of, that's interesting. Yeah, I actually, I mean, I knew about it from the ITP study,
right?
But yeah, it's a common anti-nausea medication as well. That
so
may
he's
have
already getting
geroprotective effects, and
so he's already on the protocol.
yeah. I assume the meclizine was only for a short period of time.
Yeah, it's a small dose. It's a very small.
But I mean, that's another intriguing one where from the ITP, the mechanism that was, at least how that was proposed and selected, was based on the potential that it also is an mTOR inhibitor. So same idea as rapamycin, but could be lots of other mechanisms by which meclizine is working to have an impact on longevity in mice. And intriguing to think about the possibility it could have similar effects in people and dogs. Yeah.
Well, it was a great conversation. And where can people learn more about you? Where can they find your channel?
Yeah. So at mcaberline on LinkedIn, X, Instagram, and then the Optispan podcast is, you can find all sorts of content. So if you enjoyed this and maybe got to get a little bit more edgy, you can check out the Optispan podcast.
Yeah, we'll put the link in the show notes. And yeah, thanks for coming.
Thank you.
All right, that's it for this episode. Make sure you check out my new book, The Longevity Leap, on Amazon. I'd also appreciate if you share this episode with a friend or family member. Other than that, my name is Siim. Stay tuned for the next episode. Stay empowered.